Okay, this video is the opposite of the previous video where we examined lipid storage, and now we are going to be looking at lipid mobilization from adipose tissue. So the question becomes, once we store the lipids in adipose tissue, when do we need to access them and how do we need to access them? Well, obviously this is going to be in a fasting state. When we are in a catabolic state, that means that we are in an negative energy state, and so we are going to need excess energy to generate ATP. So recall that our primary energy source is glucose. As our glucose levels begin to decline, we need to supplement this with an alternative energy source, and that becomes fatty acids as our secondary source of energy. Now, how does this stimulation work? Well, recall, here's our plasma membrane of our adipocyte. In a fasting state, catabolic, recall that the hormone is called glucagon. Glucagon, of course, is going to be elevated in a fasting state because we have low glucose levels, and so our alpha cells in our pancreas will be secreting glucagon. Now the glucagon is going to stimulate its receptor. Remember that the glucagon receptor is a 7-transmembrane receptor, and this is the extracellular hormonal signal that tells the cell we're in a fasting state. Of course, adipose tissue is going to have a glucagon receptor in order to communicate to the adipose tissue that the body is going to be needing fatty acids, so let's begin mobilizing them into the blood. Now the glucagon signaling pathway we've seen before, binding to the receptor, will activate the heterotrimeric G protein, our alpha, beta, gamma, and in doing so we are going to dissociate the alpha subunit of this heterotrimeric complex, and it will become on its own. This is now considered the active form. Active means that it is going to bind now to another protein that's located on the intracellular surface of the plasma membrane. And recall, this is adenylate cyclase. The role of adenylate cyclase is it is an enzyme that is going to be converting ATP into cyclic AMP. Cyclic AMP, of course, is our second messenger, and this will go to bind and activate protein kinase A. Now we have the active kinase that is going to be stimulating the activity of the adipocyte in the fasting state. So this is going to be occurring on a multi-step reaction. The first enzyme that is going to be activated is called ATGL. This stands for adipose triglyceride lipase. Now, from our previous videos, you'll know that a lipase is involved in cleaving fatty acids from their ester linkage to the backbone of the triglyceride, which is glycerol. So protein kinase A will induce the phosphorylation of ATGL. And of course, protein kinase A uses ATP for this reaction, generating ADP, to phosphorylate the ATGL. ATGL, once it's phosphorylated, becomes active. Now the job of ATGL is to take the triglyceride also called triglycerol, triacylglycerol, and convert this into a diglyceride. Specifically, what will be generated is 2,3-diacylglycerol. We will be releasing a fatty acid. Now the question becomes, ATGL is a hydrolytic reaction. Being a hydrolytic reaction, we have to add water across that ester linkage that links the fatty acid onto the glycerol backbone to release it. Remember, the triglyceride is found in the lipid droplet of adipocytes. This is a completely hydrophobic domain, and it is surrounded by the protein called perilipin, which protects the plasma membrane from the harm from the hydrophobicity of the lipid droplet. So how does a hydrolytic enzyme, ATGL, get into a hydrophobic lipid droplet and perform a hydrolytic reaction? Well, the protein kinase A is going to phosphorylate perilipin. And again, this will be using ATP for this reaction. And the phosphorylation of perilipin now allows it to move away from the lipid droplet. Once it moves away from the lipid droplet, now the ATGL has access to the triglyceride structure and can perform the reaction right at the surface of the lipid droplet. The next enzyme in the sequence is called HSL, and this stands for hormone sensitive lipase. Hormone-sensitive lipase is responsible for taking the 2,3-diglyceride structure and performing the next hydrolytic step to release the fatty acid. Again, it is involving hydrolysis, so water will be added across that ester linkage to release the fatty acid. And what we will be generating here is 2-monoglyceride. How do we activate HSL? Same principle as the activation of ATGL. We're going to use the kinase activity of protein kinase A, since it's been activated due to the fasting state, and we are going to add a phosphate group on HSL to make it active. And of course, like other reactions, this will be using ATP. Now the final step is that we have one more fatty acid on the glycerol structure. We don't want to waste this, so we are going to convert this into the release of fatty acid because it's a hydrolytic reaction, and we are going to be generating glycerol. The enzyme that does this is called monoglyceride lipase, sometimes abbreviated MGL. This enzyme, as far as the literature and research has shown, does not require activation. That means this is constitutively active. What does constitutively active mean? That means whenever there's a 2-monoglyceride structure available, this enzyme will cleave the last fatty acid off. Now, what happens to these fatty acids? These fatty acids, of course, are released into the blood. And they are released by passport through fat P. Recall the fatty acid transport protein is a facilitated transporter on the plasma membrane surface, and it can operate in both directions. In the fed state, we are taking fatty acids into the cell for storage, but for fasting state, we are now releasing the fatty acids from our triglyceride stores, and so they are allowed to pass into the blood. Because fatty acids are hydrophobic, they are insoluble, poorly soluble in the blood, and so we are going to couple them binding to albumin. So the largest concentration of protein in our blood is albumin, and albumin tends to be very sticky, and it binds fatty acids in a non-covalent fashion. The non-covalent binding of fatty acid to albumin allows for the transport through the blood, and then the fatty acids can dissociate when they arrive at tissue, for example, muscle tissue in a fasting state. Now, what happens to the glycerol? The glycerol will also be secreted into the blood, and the blood will transport the glycerol to the liver. Because we are in a fasting state, that means that the liver is performing gluconeogenesis. So the liver will take the glycerol, convert this into glycerol 3-phosphate. The glycerol 3-phosphate will be converted into glyceraldehyde 3-phosphate and be used for gluconeogenesis, which of course is generation of glucose in a fasting state. Now, the one final note of this is that the signal, let's go back up to the beginning here, the signal glucagon is signaling a fasting state, so that is just telling the adipose tissue that the body is in a negative energy balance, so it needs to release some of its stored energy. What happens if the body's demands increase rapidly? For example, under a stress response. Remember, epinephrine, come from the adrenal gland, is another one of our short-acting stress hormones. It makes sense that the adipose tissue would respond to stress by releasing more energy, because the muscle tissue, for example, may need it if its glucose stores are depleted. So the epinephrine will bind to its 7-transmembrane receptor, and recall that the epinephrine signal transduction pathway is the same as that of the glucagon receptor. The receptors are different, but they signal through the same pathway. So the epinephrine receptor will undergo the activation of heterotrimeric G protein, releasing G-alpha. G-alpha will activate adenylate cyclase and increase the concentration of cyclic AMP. Notice with fasting conditions and stress hormone response, we have two independent signals that are converging on one second message. You can imagine that this is going to dramatically increase the concentration of cyclic AMP and thus increase the overall activity of protein kinase A in the cell. The way I like to look at this to make it a little easier is that glucagon is going to activate a certain amount of signaling molecules to release some fat. And this is simply because we are in a negative energy state in fasting. On top of this, if we encounter a fight or flight reaction where our epinephrine goes up, that's the body's signal that we need much more energy than just simply the negative energy demands of the body in a fasting state. And so now we are going to feed into the same pathway to really ramp up the signal and increase dramatically the activity of these lipases to release more fatty acids. So it's like turning up a dial. turn the dial up on the stereo a little bit to release some fat in a negative energy balance, turn that volume up even more if we have a physiological stressor that requires more energy.